Hi, how's everyone doing? I'm Gabriel Sanchez, CEO of Inspectra Health, and I'd like to tell you about virtual biopsies. A virtual biopsy is an image of cellular structure, but taken in living tissue, so without any incision, completely painlessly. And that's a major advance from the way we have visualized cellular tissue for the last 150 years, which has relied exclusively on excised tissue. We've been developing this as a solution for early skin cancer detection because skin cancer is such a massive problem. The statistic that surprises me the most is if you take the entirety of cancer, 79% of that is non-melanoma skin cancer. It's a massive proportion, and correspondingly a large number of patients, biopsies, and dollars that we spend every year treating skin cancer. If we boil that down to a personal scale, what that means is 1 in 5 of us will develop skin cancer in our lifetime. So it could be you. If it's not you, it's probably somebody you know. So it's a major problem. Fortunately, skin cancer is readily treatable if identified early. The problem is the process is fairly complicated. So it's long and arduous and broken into 2 major phases, hinging on this question of whether or not to cut the tissue. So prior to cutting, it's a screening question. The risk of being too aggressive with the knife is that you probably cut too much benign tissue, and then the risk of being a little hesitant is you delay the diagnosis. At some point when the clinician decides to cut the tissue to get a definitive diagnosis, that starts the diagnostic process. It takes about a week, ultimately resulting in a slide, and then that can be interpreted. And what we find is the tissue that goes through that process, half of that reveals benign tissue, so that's not particularly good either. So we'd really like to change that. We'd really like to integrate this into a process that is incisionless and instantaneous and painless so that we can do this differently in the future. And what that would look like in the future is you would have something you're worried about, or your dermatologist sees something they're worried about, and we don't have to struggle with this question of should we cut this or not, and if we cut it, do we have to wait a week to know what's going on under the surface? We can just touch our device and image it instantaneously. So our device images through the cross-section of the tissue. You see cells, hair follicles, connective tissue, microvasculature, all with multispectral and multicolor rendering in a digital format based on confocal and multiphoton microscopy. So it really compresses this whole process to something where I'm concerned I get an instant digital image of the cellular anatomy. And that's important because we want to collect a lot of digital images. We're embarking on a mission to build what would be the world's only in vivo pathology database of a sufficient scale to do machine learning training for algorithms for image classification. And what that would do is kickstart sort of a self-sustaining virtuous cycle of technology improvement that would lead to earlier and earlier diagnosis. So specifically, when we reach a critical mass of images, we can do image classification and decision support that would expand the base of users that could use our technology. That would fuel in more data. We'd get more data on subtypes of skin cancers or even pre-cancers if we could predict which ones will become problematic. So that becomes an exciting sort of spiral of improvement, all fueled by proprietary data. What that means for your everyday dermatologist is not that they're actually losing biopsies. It's the opposite. They will see more spots and they can image more patients. And all of that in a completely medium-free, fully digital pipeline. And that's going to be really essential for driving adoption and integration of this new non-invasive pathway. This really positions us in a unique space. There's been a lot of activity on skin cancer screening using secondary physics like spectroscopy or impedance or superficial photos of the skin with AI analytics. However, if you really want to look at cellular structure, you have to resort to one of the biopsy-based techniques, either traditional biopsy or the emerging space of digital pathology, which is scanning the slides. So we're really the only technology that has the non-invasiveness of a screening tool, but the cellular information of a traditional slide from pathology. And that creates a really unique opportunity, some of the aspects of which I've highlighted here. For 2021, the big push is that we're pushing to complete our multi-site clinical trial. That will support our first 510(k) clearance. We'll also be prototyping and proving the feasibility of some of these AI tools for the future. Beyond that, we see this as a platform, and we've been in parallel developing different embodiments of the technology. So we have, for example, an imaging biopsy needle. So think of your traditional prostate or breast biopsy needle, but with optics down the bore. And again, that changes the whole strategy from, did I just gather the right tissue so I can try to see if I've diagnosed this properly later, to more, what am I seeing right now? And is that a problem as you look down the bore of the needle? Or similarly, intraoperatively, if you're looking for the margin of a bulk tumor, rather than shaving off that tissue and sending it to the path lab for frozen pathology, just gather an immediate image of the cellular anatomy. And you can even beam that to somebody else in the hospital or across the country. So it really sets up a new future for how we would access and utilize cellular imaging information. We have an awesome team, and we're growing, and we have big plans in sight for 2021. We anticipate we would be raising a Series B sometime in 2022. So if you're interested in learning more and you'd like to talk, I'll be in the breakout room. But that's all. Thank you.